Alright, so now we're going to talk about protein synthesis. Now we talked about DNA replication, so we have a little bit of an understanding about nucleotides and how nucleotides join together to make the ladder-like structure in DNA. Now we're going to see, and also actually we saw how that unzipped and how new free nucleotides came in and created two new strands where there was originally one. So in protein synthesis we're going to see something very similar, but instead of just recreating the entire DNA sequence, we're going to look at like specific areas where one gene is opened up, transcribed into messenger RNA, and then translated into a protein, which would then start either functioning within the cell itself or leave the cell and enter another cell and function or change a function in that target cell, for example. Okay, so in this is the first part of two. We're going to have a quick overview of what protein synthesis is, which is kind of what I just spoke about, and then look at the first phase of the two, transcription. There are two phases to protein synthesis: transcription followed by translation, and I'll go walk through each one of those. So starting with our cell, our nucleus. Now we know there's chromosomes in here, and the chromosomes are long strands of DNA. And sequences on those long strands code for protein. So gene 1 codes for the first protein on that particular chromosome. Now in green here I've drawn the DNA with the base pair sticking out. It's a bit more obvious here. You can see the DNA, the double strand either side, rungs, and then coming into the middle there's the A, C, T, G, and when it was joined together there's T, G, A, C. That's been opened up and exposed because this is the start of a gene which is going to be transcribed. Now if I was going to be smart and clever, I would have put the initiation sequence right there just to drive home how this whole thing gets started, but since we're not doing this as geneticists, we're doing it as physiologists, we're just going to see the principles of how it works and leave it at that point. So in transcription, the name tells you a lot about what's going to happen. When we script something or scribe it, we make a copy. So monks back in the 14th, 12th, 14th century, etc., before printing presses were available, would handwrite or transcribe out the Bible, and so that these copies could be sent around to the various churches, etc., in Europe, for example. So they were scribes. They weren't changing the language, they were merely making copies of it so other people could get a copy of it for themselves. So in transcription, we're not changing the language, we're staying in English. I'm speaking in English here, we're not going to change that, but we're going to make a copy of the relevant part. So for example, in a phone book, if any of you are Terminator fans, for example, you might remember that when Arnold Schwarzenegger's Terminator, the first film, arrives back in the 80s, one of the first things he does is he finds a telephone book. He looks for Sarah Connor's name, and there are 3 of them, and he tears out the page of the telephone book and takes that with him. Now that telephone book could be the whole of the nucleus and all the chromosomes in it and all the information those chromosomes carry in them. But if you just want to make one of the possible 90,000 proteins that you code for, then you just need one page of that book, and you need to open it up at that page and read that information. And this is exactly what happens on the DNA. A signal comes in and binds to a specific part of the DNA, which causes the part below it to be opened up and therefore expose the bases. Once this happens, you've then got the reading frame opened up. Now, it can be either side, which is the actual reading frame, and I don't want to get into whether this side or this side is going to make the sense and this one's going to make nonsense. We just accept that whichever side it's actually on, that's the one that will then be transcribed. So in this case, it's this side here that's being transcribed, so it's being opened up. Now instead of DNA nucleotides floating freely in the area coming in, it is RNA nucleotides coming in and matching up. Now the reason for that is the enzyme being used is RNA polymerase, not DNA polymerase, which was involved in DNA replication. So the RNA polymerase organizes the free RNA molecules to come in and line up. Now there's a difference here immediately. A in DNA binds with T for thymine, but RNA nucleotides don't have a thymine, they have uracil. I don't think I've written that down anywhere, so I'll write that down. U equals uracil. And uracil is chemically extremely similar to thymine, but it's found on RNA nucleotides, whereas thymine is found in DNA. So A will line up with uracil from the RNA, C to guanine, T to adenine, G to cytosine. And then they'll be joined at the back by the RNA polymerase, but instead of becoming a part of a new molecule, they will not join in the middle there. This single strand will float away once it's got to a termination sequence, and that will form the transcription that will eventually be translated into a protein. So that's how we get it started. So as we're seeing here, as the process goes along, you have an initiation, which is starting up here with the first base becomes available, elongation of the messenger RNA as all the new base pairs line up in complement to what the DNA strand has available. RNA polymerase joins the backbone, phosphate to sugar, to make the backbone there. That's the elongation phase. And finally, you come across a termination sequence, which causes the process to stop and the messenger RNA to come away. So that is essentially transcription. It's 3 phases. It is opening of the DNA at the specific area or specific gene, making a complement, because it's not making an A, a C, a T, and a G, it's making the opposite that it joins to, in this case U, G, A, and C, so making a complement strand of it, and then not joining to the DNA, but joining its backbone and coming away as a single stranded messenger RNA molecule. And we call it messenger RNA or mRNA because it's a messenger, it's carrying the transcript with it. Now this messenger RNA needs to be slightly changed before it can leave the nucleus and enter the cytoplasm or the rough endoplasmic reticulum, depending on what the protein will eventually do, because it can't be translated into a protein until it leaves the nucleus, because the ribosomes that are going to do it live and work outside of the nucleus. So what therefore happens? Well, in messenger RNA, once it's come off the DNA, a couple of things happens to it. When messenger RNA is formed, it's formed directly off this sequence A, C, T, G, etc., carrying on for hundreds of bases. But there are sections of these bases which actually are considered to be junk information, fillers if you like. So like having a book with, you know, illustrated with pictures, so you've got the words here and then the next page is a picture, which you don't necessarily require to understand what's being written. And so these are called introns and exons, and so if we draw a line of the DNA, I'm saying draw the messenger RNA, sorry. So we have like that, and then you have so these are the orange ones are the exons and the purple ones are the introns. So as it's before it leaves the nucleus, the introns are removed, and this becomes a shorter molecule, but these are all exons joined together. So introns are spliced out of the messenger RNA, so it's almost like a shorter version of the original message is carried out. Now, to get it out of the membrane, it needs to be capped and tailed or topped and tailed, and all that means is that for it to interact with the membrane to go out through one of the pores, it needs its ends with a little bit more information added to it, in the same way as if you go to a concert or something, you need a ticket to get in the door. You need to have shown that you've paid for your ticket. The messenger RNA molecule has to have a ticket at either end in order for it to interact with the inside of the membrane wall in order to be allowed to leave through the cell membrane through a protein. Now it can go one of two ways. First of all, if the protein that's going to be formed from this is going to work in this area here, then it's just going to leave a pore, find a ribosome, these purple dots here floating freely, and the ribosome will translate it into a protein, which will work in the cytoplasm. If, however, it's going to work in the cell membrane or possibly somewhere outside of the cell, it doesn't go through a pore, it goes through a special pore which leads directly into the rough endoplasmic reticulum, and it stays in those flattened tubes and is processed into a protein by ribosomes crushed, stuck on the outside of it. That's why it's called a rough endoplasmic reticulum, because of the presence of the ribosomes make it look rough under microscopy. So that's transcription, excuse me, and post-transcriptional changes, which is the splicing and the topping and tailing, or capping and tailing. Excuse me. We've gone through the stages of the transcription, which happens here, and now, just to remind ourselves, between DNA and RNA there are three differences. In the RNA molecule, the sugar is a ribose sugar, and in the DNA molecule it's got one oxygen left, so it's called a deoxy, one oxygen, ribose sugar. So the actual backbones are slightly different. In bases, RNA has uracil, whereas DNA has thymine, but they do the same job. And finally, RNA is single-stranded, whereas DNA is double-stranded. Alright, so that's transcription and protein synthesis, and then after the break we'll come back and we'll talk about translation and how you get the protein, and that will be us.